Benoit did his, well, I guess I should say, I think, maybe you'll correct me if I'm wrong, I think Benoit originally planned a career as a professional pianist, but then thought better of that, and instead pursued a PhD with Martin Karplus at Harvard. He then did a very brief postdoc with Jeremy Smith, and then began his independent career at Montreal in chemistry, and I think he also had a physics appointment, where he rapidly rose through the ranks, garnering the Rutherford Medal of Chemistry from the Royal Society of Canada. He moved to Cornell Medical School in New York in 2000, and then to Chicago in 2005, where he's been ever since. And today he's going to tell us about 3 different biological systems using molecular dynamic simulations that we understand them. So, Benoit.
Thank you very much. So, I'm wearing the microphone because they're filming, and so should not let my voice rise too. If it's too loud, is this too loud?
No, it's not.
It's not too loud. So, well, this is really my kind of trip to give a seminar and then go home after the visit. So this is like one of the shorter trips you can do, and this is really pleasant. So it's very nice to give the seminar in chemistry, and I've had like a great day visiting with everybody. So when Aaron invited me to give this talk, I thought like, okay, so you know pretty much molecular dynamics, you know, generating classical equation of motion and stepping in the configurational space and generating these cute movies and all that. And I thought you're probably not so interested in just methodology. I mean, methodology is part of what we do. I would say maybe not quite half, but maybe almost a half of the lab is often involved in developing some methodology or another. But I wanted to give you a sense of specific biological problems where we use the simulation to really answer as much as we could a specific questions. And so these are 3 systems. There could be a few more, but I think they are quite representative of the kind of question you can pose and try to address, and the kind of machinery you have to put in place to answer that kind of question. And so this is a potassium channel. I'm sure you must know channels by now. There's so many channels around everywhere. So they're membrane proteins that are controlling the passive motion of movement of ions across the membrane. Some channels are very, very selective, so they're called by the name of the ions that can go through: sodium channel, potassium channel, calcium channel. And so there will be some aspect of channels I'm going to talk about. Kinases, phosphatyrosine kinases, are much more complicated and kind of iffy proteins, because compared to channels, when you work on channels, they know how to behave. Kinases are very complicated. They're typically involved in all these signaling cascades in living cells, and many of them are involved in cancer when they're a bit too active compared to what they should be. You can start to have cancer. So a bit of a project about the kinase. And pumps, which are also membrane proteins like the channels, but as opposed to the channel, they don't really move the ions downhill in terms of the thermodynamic rate, and they actually spend energy. In that case, it will be the sodium-potassium pump, so it uses the energy of ATP to move ions against their gradient. So namely the sodium-potassium pump, and that's the first project. So this is work that was largely done by Aibo Yu, a postdoc who is now a professor in Australia at New Wales University. And what is the basis for the alternating ion selectivity of the sodium-potassium pump? And here's the general idea: is that this pump is a huge monster. It's a big, big membrane protein. And it has binding sites for ions somewhere in the membrane like that. And typically it's rationalized with this alternating axis. There is a clock, huh? Yes, just to make sure. Is it 4:05? Okay, just to make sure. it has many states. It's been rationalized like that when people study these membrane proteins. And typically what it does is that it's able to bind sodium from the inside of the cell, change the conformation, release them, now bind potassium outside the cell, and carry them inside. So basically it grabs the low potassium concentration on the outside and pump them inside the cell. It grabs the low sodium concentration on the inside and throw them out. And about half of our energy, we're half the energy of an animal like us. 45% of our energy is spent burning ATP for the sodium-potassium pump. So that barbecue pork sandwich today was, half of it is for the sodium-potassium pump. And so this red structure down there is the only state that we know by X-ray crystallography so far. It is a potassium-loaded state. It's called the E2 states because these are called E1, E2, ATPase, and so it's a potassium-loaded state. And so this is the crystal structure. You see the 2 potassium has 2 potassium bonds like that. There are 2 crystal structures actually. There was the first one that was about 3.5 angstrom resolution, and then after that there was another one at 2.4. So, but they're the same state, unfortunately. So we don't know so much more about that. We know a bit more because there's also a calcium pump, the reticulum endoplasmic calcium pump, the CERCA pump, where more states have been crystallized. So it's very useful as a template because the homology between CERCA and the sodium-potassium is very high, especially for the transmembrane region. And so in the case of the sodium-potassium pump, we actually have a crystal structure over the corresponding E1 state. And it's, even though this is just a homology model, so this is the E2 P state, so that's the crystal structure of the potassium-loaded state. That is a homology model made of the E1 state, and this is a sort of a morphing between the two. And if you watch the movie going, it should be, one more time, let's try that again. Yeah. So when it's bound to sodium, actually like 3 sodium are bound. And then when it's the potassium bound, it has only 2 potassium bound. But if you look actually, the ligands that are coordinating the ion are nearly the same. It's the same side chain. So when people make mutation, everything, it's the same side chain. And people have been very baffled by how can you have a molecular machine like that is able to be selective for sodium and then become selective for potassium, and yet the chemical moiety that are acting to do that are the same. Now, of course, many biophysicists / structural biologists / crystallographer would say, well, we just have to know the structure very, very precisely, and obviously if you knew this to the 10th of the angstrom, you would see the difference, and that would explain selectivity. It turns out I don't believe that's exactly the answer, and even if you did, you would say, well, are we able to explain it? So far we don't have the crystal structure of that guy, but we'd like to understand more the basis of the selectivity of that guy, and I think it's going to be illuminating. So what we did is, of course, there's crystal structures. They see the potassium, they replace them by rubidium, they know their potassium, there's no big uncertainty about that. So we made a model of the binding state using the crystal structure. In that particular case, we were not interested in simulating the big monster. This thing in the computer is probably more than 100,000 atoms when you solvate it with the membrane and everything. And since what we are asking our question about the selectivity of the ion binding site, it's not really worth it to have everything. So what we do is, we have a methodology we call the generalized solvent boundary potential, which is extracting a piece of a system like that, but everything that's outside is actually mimicking the long-range electrostatics and everything. So this has a few thousand atoms, but it basically mimics the larger system for a fraction of the cost. So for us, that was enough. And now what do we do? What is the essence of asking a question about selectivity? It's mostly about free energy perturbation. So I think this is one of the few equation slides. But essentially, you want to know what's the free energy difference between sodium and potassium in the system. So it's like the ratio of 2 configurational integral. And you rewrite the top part as being the average of a Boltzmann factor in the state A. Essentially, that's the essence of free energy perturbation. Of course, you don't do exactly like that, but it's basically the general spirit. And because you extract basically a Boltzmann factor to average the state A here, and this is just the element you're going to have. average. And so if you apply that methodology, let's say to a sodium or potassium in water, you get about 17 kcal difference, which is the difference in solidation-free injection of sodium and the potassium in liquid water. In fact, that is what you get. It's an exact agreement experiment, not because it's a fantastic model, because that's actually how you parameterize the force field. So the force field is organized to give you that number. This is how you make a sodium and a potassium in the molecular dynamics force field. And but then you want to apply that same calculation into the binding site to see if there's going to be a difference. And when we did that, IBO did that, what we find, so let me remind you that if you look at this equation, the number is negative when you're selective for sodium, it's positive when you're selective for potassium. Numbers are negative. First time we do the calculation, say, oh my gosh, these binding sites are actually sodium selective. Stop the press. You know, this thing is completely wrong. Now you could say, well, we know these force fields are bad, you know, who trusts molecular dynamics anyway, all that, you know, the normal runaround, but this is a pretty disturbing result. You know, you have a crystal structure supposedly loaded with potassium, you do the free energy calculation, it's selected for sodium, so what is going on? Well, it turns out that the ions are coordinated by lots of carboxylate groups. Do we know the protonation state of those carboxylate groups? So if you start to actually redo the calculation, so when it's in red, that means the group is negative and the carboxylate is negative. When I put it in black with the little plus sign, that means we actually protonated. And what you find is that it's only when you have a certain combination, so we did a calculation on both PDB files, one is lower resolution than the other one, but you can see you get these positive numbers, which means potassium selected, only when several of those carboxylates are protonated. And when you deprotonate the others, you basically lose that selectivity. We've done also some calculation with mutants, like for example, you could say, oh well, a protonate, like a reviewer said, a protonated carboxylate is basically like an asparagine or glutamine, so why don't you do the calculation? Turns out actually glutamine is not a protonated carboxylate, it's a bit different. And so you can expand that table to have numbers where you have mutants, and they're not necessarily on par with the protonated groups. But what you can see is that, okay, so these sites will be selective if there's enough protonation of these carboxylate groups. And you can also look at the fluctuation of the simulation, and you can see the thicker line here. The thicker line is actually the crystal structure. And the deviation from the crystal structure when this is a protonation group, you see, and here as well the glue with the OH, you see here, a glue 334 with the protonated. The deviation from the crystal structure is minimal when these groups are protonated. If you deprotonated them, the deviation in the crystal structure, in the dynamics compared to the crystal structure is much bigger. So that's comforting, number one. Number two, now the sites are selective when they're protonated. Number three, we also did some pKa calculations, which can be done at different levels, and it indicates that indeed those groups should be protonated. So in our camp now, we did everything we could. What can you do more? Well, now you go talk to an experimentalist, because you'd like to see whether that's possible or not. So Pablo Artigas is a friend of ours, and he's a good experimentalist on the pump in Texas, and a pump is not a channel, but because it pumps ions, so these are macroscopic measurements, but because it pumps ions across the membrane, you can actually measure an electric current. You give ATP and you measure the current, and that means the pump is working. You can see the pump is working, but you see, these are cycles where you change, every one of those cycles, what he did is he changed the concentration of potassium on the outside. If you increase the potassium concentration on the outside, the pump has to work much harder now, because remember, it's trying to take let's see, no, I'm confused. Let's see, I almost misspoke. Yeah, no. If there's lots of, it's trying to throw away the sodium on the outside. If you increase the potassium concentration, it will have a harder time throwing away the sodium. And it's actually the contrary. I'm sorry, I'm getting confused about that now. Yes, Yes, so it's working harder when you have little potassium, not lots of potassium. I'm sorry, I'm confusing you. So you can do that experiment at 2 pH. And you can see how hard the pump is trying to function. And you can see what is the maximum current you can generate across it. So what you see here is the plot here is like the half concentration of the maximum current. So if you have a low pH, at pH 6, the concentration of potassium to make the pump happy is pretty low. It works well. If you go to pH 9.6, tells me now these carboxylates are deprotonated, you have to go to a higher potassium concentration to make the pump happy. So that's logical. But is that enough? So what you can do then is take away the sodium and put instead NMG, which is a substitute, and other ions, but it won't work in the pump. And now you play again this game with the pH, and you see that there's almost no difference anymore. Which means that it was because this big difference between this solid line in black and the red line here is because potassium is trying to fight against the sodium, and when you change the pH, it has a less a good time to fight. When you remove the sodium, the fight is no more necessary, and so that's why that difference. So one possible explanation of these experiments is the theory that we propose, that actually these binding sites require potassium to be selective. It's not necessarily the only one, because that's a very complicated protein, and it could be that the pH actually affects some conformational change, the balance of conformational change. So it's, unless somebody really does like a super high resolution structure, maybe with neutron scattering where you see the hydrogens into the binding sites, you won't really know for sure. But in our hands, that's about as close as we can be to confirm the calculation. What we think happens when you go to the other state that's sodium selective is that these protons are not there anymore. Because the binding site will be much more prone to be selective for sodium if there's no proton. And so could there be a kind of a back and forth of proton binding to these groups to make the site potassium selective, then the protons go away and then the site becomes again sodium selective. And so there could be a kind of a conjunction of proton motion. We know that the pump does not pump protons, so the protons cannot have a net flux in one direction. But there are leaks of protons, so it could be that protons go back and forth across the membrane. And the other thing is that in Cerca is actually a proton calcium pump. And it turns out that the state where we think the protons are bound to favor the binding of potassium is known to be the Cerca state, which is protonated. So this is it's a nice combination of evidence that points really towards the fact that probably protons are involved in the real mechanism of the sodium-potassium pump. So that's one story that was published. That was published in Nature Structural Biology last year. The kinase. So the question we're trying to answer here is, Gleevec is an inhibitor of kinase, and why is Gleevec a specific and potent inhibitor of Abel kinase, which is involved in leukemia, but not of similar kinase like CSAR kinase? And this paper is, I would say, optimistically in press at PNAS, even though we haven't heard from the editor. Optimistically, I'd say it probably is in press, or accepted for publication. Wei Jian also was involved in many of those calculations. So what's the story here? So the kinase, this is Abel kinase. It serves to transfer the phosphates from ATP onto other protein, other tyrosines, so it's a signaling protein. And when the kinase is overly activated, you can get leukemia. In the binding pocket, there is a part of the protein that's part of an activation loop. It's called the DFG motif. DFG for D-aspartic, F phenylalanine, and G the glycine. And that motif has two conformations. There's the DFG in that you can see like that, where the phenylalanine is sort of in the screen. And the DFG out, where the DFG, the F is out of the screen. And Gleevec has been discovered in the 1990s as being a very potent inhibitor of Abel kinase, and John Curien got the crystal structure of Gleevec bound to Abel. And the first thing they saw is that when Gleevec is bound to Abel kinase. It's actually bound to the DFG out conformation. This is Gleevec bound like that. And when this was found, it was the first time that anybody had observed a kinase with this DFG out conformation. It has never been seen before. There's crystal structure of kinase, pKa and other Src kinase for a while, but the DFG out conformation had never been seen before. And so that led to the hypothesis that perhaps only Abel is able to have this DFG out conformation, because Gleevec is not a potent inhibitor of other kinases like C-Src, which is a kind of a grandmother of all kinases. And so they thought like maybe the specificity of the drug actually arises from a conformational selection of the proteins. Like there's an accessible conformation and the drug binds to it, but other kinases, they don't do that. So that was for a while the question. It's disturbing though, because C-Src and Abel and all the member of the Src family are extremely homologous to each other into sequence-wise. I mean, Yen-Lin, you can probably correct me if I'm, is it like 70% similarity, the sequence, 70, 75%? It's very, very high. Yeah, so close to the binding site and the loop and everything, the similarity is very, very high. And so it's kind of odd, and in fact, later, again, John Curie got the structure of Gleevec bound to C-Src. Now, to get that structure, they thought it doesn't bind, and now they get a structure, but they had to crank up the concentration of Gleevec quite a lot. But nonetheless, they got a structure of Gleevec bound to C-Src, and lo and behold, it was DFG out. You can say, oh, well, so much for that, I guess. So it can adopt that conformation, but it's still a fact that Gleevec binds less well to C-Src. So you could say, well, the DFG can go out, but it costs more to do that, so hence you don't bind so well. So it binds very much in the same conformation. You see the red is Abel and the blue is the Gleevec of C-Src. You can see that they bind exactly in the same conformation. The DFG out is the same conformation. It's very, very similar. So people say, well, perhaps it's because there's an extra cost to have this DFG out conformation. But there are other people who say, well, we don't believe that. We believe it's more like the drug has just an affinity for that pocket, given the sequence of amino acid in the pocket of Abel versus the amino acid in the pocket of C-Src. Yeah, and then so that raised the issue of, is the selectivity or the specificity of Gleevec for this kinase coming from the conformational selection, basically the DFG in or out, or does it come really from the affinity of the drug for the binding pocket? And this is really not so easy. Potentially, somebody could try to answer that experimentally, but that kind of dissection of binding is very, very difficult to do experimentally. And so this is a good place for computation for trying to clarify the matter. And in order to do this, well, you have to do the whole enchilada. You have to compute the absolute binding of Gleevec to the pocket and try to see what are the different contributions. And so first we have to deal with the conformational selection, because this is a conformational change. You can see a few of the amino acid. It's in the interior of a protein, so even though it's only DFG in, DFG out, it's actually not so trivial to compute that conformational change, the free energy for the conformational change, because it's at the interior of the protein, and moving a phenylalanine across the protein is not so easy. So this was very much the work of Yilin Meng, who's sitting in the audience here. So what we did is first use the string method, which is a method to discover pathways, to go from one conformation to the other, so that you sort of find at least a plausible way to go across. And this string method is basically you start from like a path. This is a cartoon as an example, illustrative example. You start, let's say, for this black path. And then what you do is you run simulation at every one of those black dots. Every one of those black dots is actually an entire copy of the system, so it's a full molecular dynamics simulation. And you look at the mean drift of each one of those simulations. So every one of those black dots is now shifting around, and then you correct them and then regenerate. So if you iteratively repeat this process, you converge towards a better path. It sort of relaxes the system towards a better path. And so once you have the path like that, we could then bootstrap our way to do umbrella sampling and get the full free energy surface. So, so basically, Yilin did the string method on Abel and then CSARC, and this is the DFG. So the way it was done, I think, is on pseudo-dihedrals that are applying to these sets of atoms that I'm showing here. These sets of dihedrals. So they help you see the conformational change. And then, so the initial path in black, I believe, was a kind of targeted dynamics. And then these color paths are the relaxation. And you can see the black line sort of relax towards this more plausible saddle point. And this is umbrella sampling. So this was a relatively costly umbrella sampling for this process. But then you can see the two conformation, you know, the DFG in and DFG out, and you can see they're the same color, and namely because they are almost the same free energy. This is for Abel. And the cost, actually there's a little bit difference, about 1.4 kcal per mole between the in and out. We did the same also on CSARC. And so you have here in red is Abel, so this is the DFG in and DFG out, so 1.4 kcal per mole. And in blue is CSARC, and you can see this is in and this is out. And so out is much more expensive in CSARC, indeed, by about 5 kcal per mole. And actually some people did some metadynamics using, I believe, the amber force field. So it's a different method, different force field. It was published, and they also got like about 5 kcal per mole for that DFG flip. So this seems to be a pretty robust outcome for this calculation. So indeed, there is a conformational selection for the DFG. But then we did a bit more. We want to deal also with the binding affinity part. And so this was done in large part by the alchemical free energy perturbation method, where you start from the ligand in solution and you bring it into the pocket by a non-physical process where you decouple and recouple it. And so the way we do this is it is a stage-wide, step-by-step method where you decouple, you apply many restraints and stuff like that. You literally bring it back, and then with restraints, and then you free it. And so all these steps are rigorously applied like that. And the way we do this now is, so it gives you lots of terms that you can do with the coupling and everything. And what's new here that was implemented in large part by Wei Jiang at Argonne is that every one of those thermodynamic steps now is actually coupled through a Replica Exchange. So we can run this calculation all at once, where every conformation for every coupling is actually exchanging with each other. It's mostly beneficial for the early coupling steps, where you begin to grow the molecule into the pocket, because now you're really pushing things out of the way, and that's, you're expelling water molecule and everything. And so that is always a very difficult step in free energy calculation. But now, because we have Monte Carlo exchange, this part is actually much better sampled. And so this can run at Argonne on the blue gene with something like 32 racks. I mean, you can really scale this up, because these free energy calculations really are parallelized trivially in some ways. And so Yen Lin ran that calculation. The way we run the calculation, we run it for a phase, and then we write all the outcome, the coordinates and everything, and we run it again and again. Rather than running a single long calculation, we like to restart the calculation like that. First of all, it makes a job that actually can run on the blue gene. And also, you can see the evolution, the progress in the calculation. So you can see the Abel was pretty much converged pretty fast. There are some fluctuation between the cycles. That's just because of the finite sampling at every one of those cycles, but it gives you a sense that we know pretty much that this is around, you know, minus 10.8 if you average the last steps. In the case of CSARC, it was a little bit more difficult to converge to that, and there are reasons for this. For example, we had to build some of the loops. I think some residue are missing in the crystal structure. We had to build them. And so there was perhaps more uncertainty at the beginning and needed for equilibration. But nonetheless, we get this. So you can see that, again, these two steps are indicating that Gleevec has a higher affinity for the pocket of Abel kinase than that of CSARC once they are in the DFG out conformation. So So in the end, if you compare with experiment, I'm going to skip that. When you compare with experiment, our calculation for Able gives us minus 9.4 kcal total. That's confirmation plus affinity. Experimentally, the numbers are between 10 and 8. So this is not so bad. In the case of CSART, we got something like minus 1.4, so we think we're a bit too weak, even though experimentally it's between 6 and 4 kcal per mole, the binding free energy. So it's possible that there is more uncertainty on those experimental number than this one, because it's a higher affinity. It's possible we underestimate the binding, but clearly the specificity of Gleevec for these kinases comes both from the conformational selection and the affinity. So the take-home message is somewhat frustrating for people, because people like when you have a very clear-cut black and white story. Unfortunately, in that case, it's not that simple. So it's both the conformation and the affinity. But this is important information. If also the fact that the results are in pretty good agreement experiment, and we have other calculation on other kinases where we continue to be in good agreement experiments. So it shows that this kind of fairly detailed calculation is probably very informative. People are trying to design kinase inhibitor for cancer, for example. If you're really close to the final design phase and you ask small atomic detail about these inhibitors, we could participate to help. And so the last story is actually submitted. It's under review, so that one is not published. It's very recent. It's mostly the work of Jared Ostmeyer, who is sitting also in the room. There are other participants also, of course, and their name will come up. And so the question is, why does recovery from sodium inactivation potassium channel take so long? And so these are potassium channels. They have many parts, potassium channels. The selectivity filter is the narrowest part. It's the place where the ions are typically dehydrated and they talk directly to the protein. And there are larger parts like these helices. That's called the activation gate. Essentially, the channel can be closed, and then when you send a stimulus, this gate opens. So this part opens up, and then the ions can flow through. And I think I have a little bit more. So if you activate the channel, basically it's closed, you activate it, it's open, you get a boost of current. This is a macroscopic measurement done on the KCSA channel. Now what happened is that very often in many channels, the current will sort of decay like that. And why does it decay like this? Well, it's called inactivation. You still have this stimulus, but somehow the channel is inactivating. It doesn't want to be active. What do we think is going on? Well, you have a closed channel, you open it, and it's able to conduct. The filter is conductive. But it makes a conformational change towards an inactive filter, so it's blocked. And this does not conduct. And the only way you can recover now is to close the gate, basically remove the stimulus, close the gate, and now the channel will be closed inactive, and it will take a while to recover. So going from here to here is the recovery period. Now, this channel is, again, poised to conduct when you're going to open it. And when you do macroscopic measurement like I showed before, what you see, the cartoon is like that, is that you have a channel that was closed for a while, so it's poised to go. You give your stimulus, it produces a lot of current, but as time goes by, the filter inactivates, so the current goes down. Now you close the channel, so you're here, you have a closed channel, but the filter is still inactivated. Now if you wait a period like that, you might be able to open it again and it will conduct again. And this time is called the recovery time. If the time to recover is too short, when you give the second stimulus, the channel will not conduct. If the time is very long, the channel will be ready to go. And so experimentally, it's very easy, or it's very possible to probe that recovery period. So now more about, so this has been measured, and this is experiments done by Sudha Chakrapani, who was a postdoc in Eduardo's lab. She's now assistant professor at Case Western. And these are recordings where you simulate the KCSA, open it, it conducts. Yeah, we're good, okay. It conducts, and then you can measure, how fast it will recover. And you can see that in about 6 seconds, it is able to recover. That's the kind of experiment she did. And so it's also sensitive to potassium. These are little facts, you know. So if the potassium concentration on the outside of the cell, essentially the extracellular side, has lots of potassium, the channel recovers faster. If you don't have a lot of potassium, it recovers more slowly. So when you go to high potassium, it recovers in a time that's shorter. When you go to low potassium, it takes longer to recover. Now, you're a chemist. Imagine, look at these times here. These are seconds. Huh? You're talking about 8 seconds, 12 seconds, 16 seconds to recover. Now, what is going on? Well, there's lots of crystal structures now of these channels. I drew these cartoons before, but it turns out that actually we have a crystal structure of every state possible. There was a heroic effort by Eduardo Peroso to obtain the crystal structure of the channel in all these possible states. And they got the structure of the channel in the inactive state, in various inactivated states. So what we do is we have the crystal structure of the open inactive state, we have the crystal structure of a closed channel, which is inactive. So this is in gray is the inactive open channel, and in color is the closed inactive filter. And you can see they're identical. So we know everything we need to know. The problem is, if you compare the active and inactive channel, this is the 1K4C is the 2001 crystal structure that Rod McKinnon got, the high resolution, and this is the pinched inactive filter that they got. So this is the difference between these two structures is 0.7 angstrom RMS. If you go from that structure to that structure just with charm and vacuum, just minimize the energy and go from it, there is no barrier. I mean, basically it's a high energy, it slides down to this other guy. 16 seconds, if you apply iron rate theory, you would have a barrier of 15 kcal per mole somewhere there, in a naive argument. There's not even the glimpse of a shadow of a barrier. There is nothing. Just to put things in place, 16 seconds, a ribosome of a bacteria is able to add about 10 amino acids per second. KCSA is 160 amino acids. 16 seconds is enough for a bacteria to synthesize a monomer of KCSA. This is a bit of a baffling time scale, one might say, you know. I mean, it's absurdly long. 16 seconds is ridiculous. When you start to say, well, 16 seconds to move 0.7 angstrom RMS, I mean, this is barely thermal fluctuation. How could it be? So, perhaps one of the first time actually we went on a fishing expedition, we thought like, we really don't know what's going on. Let's run a simulation to try to understand.
She's so scared.
What's so scary?
This picture.
What?
Just tell me why biology is so scary.
It's the first time that you see. This is the membrane, this is the KCSA that Jared ran. So we got time on the Anton computer in Pittsburgh. And so this is the simulation was running on Anton. Anton is the fastest molecular dynamics hardware you can use in the world. You can run about, what is it, like maybe if you have the Anton for 24 hours, maybe you run like a couple microseconds.
6-7 microseconds.
6-7 microseconds per day. When you get an allocation Anton in Pittsburgh, they typically give you a week of computer time. So you could run some time, you know. If we wanted to reproduce the 16 seconds of recovery, though, it would take us about 7,000 years. So it's still not there. So nonetheless, you know, we started. So we built the model of the closed KCSA, inactivated, and asked ourselves, well, what happens? And this is time in microsecond. We're going to avoid to 17 microsecond. In green here are ions that are inside the cavity, are poking into the filter and binding back to the cavity. So this is the S4 binding site. Here is ions that are in the extracellular side, and they come in the binding site and they leave. They come in, they leave. Not always the same ions.
C is the position.
C is the position along the axis, yeah. And you can see none of the green line actually crossed. No permeation event for 17 microseconds. So that channel is not conducting in that state. This was big news because I did not anticipate it would be like that. This distance here is the distance between the 2-carbon alpha of the glycine where the channel is a bit pinched. That's where the place where there's the biggest difference between the conductive and unconductive state. So you see, it seems to be damn stable. Excuse my French. Sorry? I mean, nothing changes. It's the most boring simulation. The most boring simulation in our lifetime. And nothing happens. But then we're so happy nothing happens. Because you see, well, what is going on? So Jared looked at this and said, you know, the most stable thing that we see is there are these three water molecules that actually are observed in the crystal structure of 2001. This is a histogram of their distance during the simulation relative to their crystallographic position. You can see they move, actually, I'm sure you can see it. This is 1 angstrom. So they move about half an angstrom away from the crystallographic position. So they never move. You see, this is fascinating. You can look at the occupancy. 97% of the time you have 3 waters in there. Sometimes, you know, you have 2 waters, like maybe less than 1%. It's really very, very low. So they're almost always there, you know. So Jared tells me that, says, remove them. Let's try removing them. Say, should I remove them? And one by one, say, nah, remove all of them. All of them. Boom! The channel recovered in 25 nanoseconds. You see, it was inactive, it becomes active confirmation. This is at 1.2 nanosecond. This is pinched. Boom! 74 nanoseconds. It's conductive. These 2 waters actually are also in the conductive conformational state. So you say, well, beat me. So when you remove these waters, the channel recovers? What is that? So we wanted to understand energetically what could be happening. So now we're going to back off and resort again to umbrella sampling. Say, this pinching coordinate is how much the channel is inactivated. So when it's like 5 angstrom, the channel is pinched. When it's 8 angstrom, the channel is conductive. And we know that the binding of an ion is important, so we're going to deal with the position of an ion from outside to inside with the z coordinate. So first thing he did is do the PMF when there's no water. And this is a potential mean force. This is the pinching coordinate, and this is the z. And just to put your idea, so this is the ion is in the binding site here. There's an ion solution. Here now the ion is lower, it's in the binding site. Now the filter starts to unpinch, and now it's happy, and even another ion has come in. This is another state where you try to unpinch a bit too early before the ion has come in. So you see, and if we actually take the trajectory I showed you before, where the Anton trajectory where the channel recovered, this is what happens. This is the trajectory. Whoa, 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 whoa. This is.
These are all without water.
Yeah, and the black line here is the Anton trajectory. So it was jiggling there, it just fell there. So the trajectory actually followed the free energy map. Then you say, well, what happened when you have the water in there? Let's put back the water. This is the PMF now, you see. So when I'm outside, but now I have my 3 waters, it's I can bind an ion. This is possible. This is the ion is bound on one of the outer side. But I can't pinch the pore. I cannot unpinch it. I try to unpinch it, the energies go high. Because why? Well, I am bumping into these water molecules, you see. If I go from active to inactive, there's a clash of the carbon alpha or the glycine 77 and the water molecule. It just will not do it. And so the 3 waters sterically are stabilizing the protein like that. So, well, is this enough? I mean, that sounds like a nice story. You say, well, yeah, but we're talking about 16 seconds here, my friend. Huh? Yeah, your little 3 waters, is that enough? So, because it's a bit baffling. You see, like, gramicidine, for example, in a channel I've worked on for many years, the pore is 25 angstrom long. And it takes about maybe 10 to 50 nanoseconds for water to diffuse from one end to the other. Here you're talking about 3 waters. It's a pore about 10 angstrom long. So, you know, what's going on? So it's actually very, very rare that the waters leave. So what we did is, and Jared did, is that you just remove the water from one subunit of the channel and you watch them rebind. You watch them rebind. And essentially he did that, I don't know, 25 times, that's here. And we did a histogram, and basically they rebind in about 90 nanoseconds. They come back. So that's about the time it takes. if you look at the very long trajectory from Anton, you'd say maybe the estimate of these waters leaving is about once every 10 or 20 microseconds. If you do the ratio of the on and off rate, that would give you that only 1% of the time the water have left. 99% of the time the water there. It is what it is. Okay. So you say, well, but there are 4 monomers. So in order to recover, you have to lose the water in one monomer, and then in the other monomer, and in the other monomer, and the other monomer, and then you have to rebind the potassium. So I made a Markov model of that. So the first one, the rate is 4 times, because any one of those 4 subunits could actually lose its water, and here only any one of these 3 could lose it, and here any one of these 2. So you make a little Markov model. You say the rate for losing the water is about 11 microseconds. The rate for rebinding is about 79 nanoseconds. The rate of binding the potassium is about 12 nanoseconds at 150 millimolar, and since it's bimolecular association, it's roughly linear proportional to the concentration, so I make it like that. You run this, and this is what you get. The star are the Markov model, and the colored dot are the experiments. So it is possible to get 16 seconds out of that Markov model. So that means that the link is pretty nice. Then you say, well, can we do more? Well, you say, you recover faster if the waters are not in the way. Could we remove them? Well, if you put a lot of osmolyte in solution, you're going to suck out the water. You're going to dry out the cavity, if the cavity is not accessible to the osmolyte. So you put big sucrose in solution. So Suda Shakapani did the experiment. So with no sucrose, you recover like that. With sucrose, you actually recover faster. In fact, you inactivate even slower. You can see here the lines inactivate more slowly than this, but you recover faster. And so that is about as close. And then I applied the Markov model on that. And what this seems to indicate is that now, instead of the cavity being empty only 1% of the time, it's actually empty 2% of the time. Which is, it seems like a small effect. You say, Jesus, we go to 2 molar sucrose and we go from 1% to 2%. But it's like, osmotic pressure it's not that effective, and the cavity is pretty small, so the PV is pretty small, or the pi V is pretty small. So it is actually all self-consistent, that when you add the high osmotic strength, you dry the cavity only so little, but now you recover faster. By the way, there is no biophysical conformational change that could actually go faster when you increase the viscosity. There's nobody can think of any alternate explanation. If you go to 2 molar sucrose, think of any molecular machine you put in 2 molar sucrose, I bet you it's going to go slower, not faster. There is no way it's going to go faster. So this is about as close a confirmation that we could get of our conjecture. So there's also somebody who measured the hydrostatic pressure influence. Stefan Heinemann from Germany measured that 13 years ago. This is an obscure paper or book chapter. I don't know where you found that. Is it a book chapter or a paper?
It was a reference in book chapter.
Yeah, but it's amazing. It's like this. It was done in a Max Planck, obviously, because you don't measure a thing with hydrostatic pressure on channels normally. And they did find that the activation volume was 255 angstrom cubic, which corresponds to 8 water molecules, which is basically the difference between this and this. See, 3 times 4 is 12. Here you have 4, so it's 8 water molecules. It's like exactly spot on, unbelievable. So seems like this is probably on the right track, that explanation. So these are 3 stories, and I think the take-home message, I could give you a few more, but I think that what's important to realize is an application of molecular dynamics and computation. What I mean by molecular dynamics, I mean also slash free energy, umbrella sampling, string method, or what have you. But any kind of detailed atomistic study of these biophysical systems starts with a well-defined question about the biological system. These are not really black box, turn the crank type of operation. You have to have a very incisive question to begin with. Then you can put the machinery in place to answer that very specific question. it really helps to have a link to experiments and to compute things that are measurable directly, or to do a calculation that actually outputs the quantities that could be measured, then they become prediction, and back and so forth. So that's important. I think it's very risky to have lots of detail in these computations that are impossible to verify experimentally. So it's important to have something that is hooked to experiment. A lot of what we do is expanded by free energy method and umbrella sampling. That's why, for me, I sort of wrap it together with molecular dynamics, but I would never think that brute force MD is the way to go all the time. It's just you need all these tools. To my own surprise, I would say non-polarizable current force fields are actually good enough that you do find meaningful answer to well-defined questions. That doesn't mean that you cannot improve the force field, but the force field are not a disaster. I think when you see a disastrous calculation, it more or less probably because of lack of sampling, or the underlying assumption about the system are just wrong. Like you thought these two proteins were together in that situation, and they were not, or something like that. But the force field is not really to blame at this point. So we like long Brute force simulation when we can have access to them, but in fact here the longest simulation, nothing happened, you know. And so it's kind of a strange situation where in fact but without the 17 microsecond, I might have not believed that what we had was really an inactivated channel. So we didn't need to have it for millisecond, but even 17 microsecond made that the situation pretty convincing. And but certainly we are strong proponent of bias sampling methods, because I think you can go much farther with that kind of technology. So I'll be happy to answer any questions. Thank you very much.
So Benoit has already followed you to answer questions.
So the long trajectory, unless I missed it, it fit into your PMF, or the main part of the PMF perfectly, right? It didn't. What
Yeah, if you
information did it give that you
sorry,
didn't get? What information did it give that you didn't get from your PMF, your umbrella thing?
So the anecdote is the following: is that 2 years before, Albert Pun is a co-author of that story, because now he works at DE Shaw, but he was postdoc in the lab, and 2 years ago. He tried to do the same calculation. He did only the PMF without the waters. What happened is that I said, oh, let's simulate the closed pinch pore. And so he lifted the PDB file from the protein data bank, but he took only the protein. He didn't take the crystal structure from the water. And so without those waters, the PMF was showing that the filter does not want to stay in that conformation. Of course, if you run it long enough, the water will rebind. But when you do umbrella sampling, I mean, the water rebinds about 100 nanoseconds. When you do umbrella sampling, you don't typically run every window 100 nanosecond. That's like not, I mean, you could, but it's not necessarily the most, I mean, 3 years ago we didn't definitely do that. And so that was mostly a failure. And when Jared started to do this on Anton, I thought he's going to simulate it, and tomorrow he'll come crying in my office saying the channel fell apart, it's not staying like that. And every day I would ask him, so what's happening? He'd say, well, it's staying put, it's staying put. And somebody said, what do you mean? It's still stable? How can it be? He said, well, yeah, it doesn't move. It's like, well, you must have something different than Albert Pun. And we had the PDB from Albert Pun, so you say, look at the difference. What? And then it's Jared who noticed that he didn't have the waters. So it's a bit serendipitous. I mean, but these crystal structures have been there for 11 years, and nobody ever mentioned them like that. They've been known to everybody for 11 years.
So how are we going to know if you don't have the water in the crystal structure? I know you've worked a lot on this. I mean, this is in my group we work on these. It scares us to death that there's water that we don't know is in there. And so what are we going to do?
Well,
you have the answer, don't you?
You mean like you have to do grand canonical multiplication?
Well, that's the fear, right? I mean,
no, but I would say, you know, if you get a protein structure and you simulate it, and it doesn't want to stay put, let's say in 10 to 100 nanosecond, it doesn't want to stay put. Yeah, I mean, if it's a crystal structure below 3 angstrom resolution, and it doesn't stay put, you either have misassigned protonation state in there, you're missing a ligand, or you're missing waters in the structure. It's not possible that the force field is so wrong that the structure would be that unstable.
Yeah.
And so, very often we have had like structures that were just not going to stay put, and probably waters are missing. You could search the cavities and try to very carefully, heal and cure that system. But,
So what is going to make, in a real living organism, what's going to make those waters go in or out?
well, so the thing is that this is a very interesting question. Actually, so inactivation in our life, in our body, is very important because these channels are an electric switch, and the inactivation is a kind of a refractory period where you can't excite them anymore. You have to wait for a little bit. And that kind of length scale, or time scale is, well, it's very, well, yeah, some of them, KCS is pretty nasty, but, shaker might be 500 millisecond, which is in our brain. But HERG is probably, in our heart is faster. So, how the excitable tissues are tuned is very, very much influenced by the length of the inactivation period. So this is of immense consequence for biology. It turns out that if you look at all the potassium channels sequence and you build homology models, the cavity to bind these waters is present. So we suspect highly that these waters bind there. But this is perhaps, the KCSA is a bacterial channel. And we speculate that, well, bacteria are exposed to very, very drastic environment. And in the gut, in the soil, all sort of stuff. And so to have a swing of, 20 molar concentration of osmolyte for bacteria, that's probably everyday life, And so it's possible that bacteria use this as if the water is a ligand that gates the channel. And so bacteria act as a ligand-gated channel where the ligand is the water. And we're just living with sort of the fossil remnant of that the water still binds, but now you have mutation to sort of tweak it back to 500 millisecond, but it's still, the echo of that old past.
Something has to be the chemical activator to move the waters.
Well, I don't know. I mean, the Markov model just says it's the probability at some point they're going to get out. They will get out. They will get out. Ultimately, it's just randomness will make them go out. Joe and Keith.
Yeah. I think you just said the key word, it's cavity. You keep focusing on waters, but look at it the other way around. What you in fact have to do is to generate a cavity of a particular type. Okay. If you look at it from that point of view, let me ask an experimental question. Has anyone looked to see what the influence of high pressure xenon on this latency period is? Because you would expect that these cavities will be big enough to accommodate xenons, which are, after all, often crystallographically detectable.
It's an interesting concept, yeah. I mean, maybe Stefan Heinemann can take this over at the Max Planck Institute, but I'm not sure doing electrophysiology with high pressure xenon is trivial. You can ask Pancho or Eduardo, but yeah. I mean, the other thing we tried to, maybe some synthetic chemists much better than we are at that, but we tried to imagine, is there a kind of a semi-nonpolar ligand that can hydrogen bond that would actually take the shape of 3 water molecules, would stick in there? Because then that would become a channel inactivator. If you had like such a substance, such a compound, you would just throw it in there and it would inactivate the channel right away.
If I may, another comment is that, chemistry is fast, but biology can be slow. I mean, the turnover number of your kinases, for example, is often, 0.1 per second or something.
Yeah.
So something is very slow. It's not the chemical steps, it's the protein conformational changes. We work on a photoreceptor. It takes 2 seconds for reisomerization to occur. The isomerization step itself is probably occurring in sub-picoseconds, but in fact, the relaxation of the protein in that case is again in the seconds domain. So I don't think you should be surprised by 16 seconds. I think perhaps one should be asking, how do some other conformational changes occur so fast?
Yeah, I guess the 16 second was sort of shocking to us in the sense that when you look at the 2 conformational states for which we have high resolution structures, they're so similar. We couldn't possibly rationalize how could it take so long to go between two conformations that are so similar. You know, I mean, like the sodium-potassium pump pumps about 150 ions, does 150 cycle per second. It's burning ATP and pumping 3 ions one way to the other way. So, you know, to have like just this poor little filter that has to unpinch by 0.7 angstrom, and it takes tens of seconds to do that, it's just, we couldn't make sense of that. It didn't make any sense. Joe had a question?
I was wondering if there's an obvious place to make a mutation in this cavity to knock the waters out, and you can test your model.
Well, Jared looked into that. The danger is that, you know, don't forget that when the channel is conductive, the cavity actually disappears. So if you make a mutation there, you don't want to actually fill the cavity with your other guy permanently, such that now the channel does not inactivate, or does not become active. So it's not so easy. I mean, almost every mutation you can do in the pore domain will affect the balance of active, inactive. And we're going to get into these kind of calculation now, but it's not so okay. I think, you know, the osmolyte thing was probably the most standoffish thing you could do, because you're really not messing with the channel. You're just changing the thermodynamic force to drag the water out. So it looked like the least intrusive thing we could do.
So related to your point about a molecule that might mimic the 3 waters, if you take formaldehyde,
Yep.
it will hydrate. So you'll have essentially a carbon atom that has 2 OHs on it, which won't be exactly like 3 waters, but it's 3 atoms, 2 of which would be OH. So it could be interesting to...
We could try. I mean, we could try to dock it in there. Yeah. There was something over there?
Yeah, I was wondering, in the crystal structure of the active state, were waters resolved in the crystal structure of that active state?
The active?
Mm-hmm.
Yeah, the active states, there's 1 water per monomer.
So people could just have looked at the PDB of the crystal structures for the active state and the inactive state.
Yeah.
Purely looked at the waters that are there and seen that there was that difference.
You know, that's often in life, is like when you know where to look at, it's pretty much staring at you in the face. But, you know, the paper by Replica Exchange 2001 is not one peep about the fact that these waters are like that. And, you know, many people have simulated that system in the last, I'm talking about 10 years now, this is not like recent. Mm-hmm. And it just didn't,
right.
Nobody thought of it this way, you know. I mean, to be fair, you know, these waters are not bound so strongly. Like, if you look at the enthalpy and everything, their binding free energy is about 1.5 kcal. But it's the concentration of water is so high that, you know, you're basically 99% of the time filled in the cavity. So, you know, nothing is really insanely crazy with this. It's just that this is how it is. And once you have 4 subunit, then the probability sort of multiplies together, and then the time scale just becomes enormously long, you know. Because to go from 10 microsecond to 16 second, it's a big leap. Even though I did nothing, I just made the Markov model. There's nothing to stretch the time artificially. Nothing at all. Yeah. Just last question, maybe.
Go back to the sodium-potassium pump. So what makes this chamber to be, those residues in the chamber to be protonated?
What makes them to be protonated?
What makes them like to be protonated? Because it seems like to be 3 of them.
Yeah. Well, we did pKa calculations, and basically on the electrostatics, the local electrostatics, they want to be protonated, a few of them, yeah. Well, the thing is that when you have a bunch of carboxylate together, chances are that some of them are going to be protonated. You can't have, like, you know, 4 or 5 negative carboxylate together at some point.
Would, like, the sodium have to carry the protons with them, or carry protons out with them?
Well, the pump does not pump protons, but, there's no net flux, there's no change of pH because the pump is working. But there are many experimenters who have reported leaks of proton current. So when the pump is working, you can notice that if you have a leak of proton, and people have talked about proton wires and proton pathways in the protein. So, you know, this is a bit in the air, I would say, but it's not completely a finished story. Maybe when we get more crystal structure of the sodium-potassium pump, not just homology models of CEFCA, but, you know, that will be very interesting too.
Okay, well, Benoit is around on campus in case there are more. Otherwise, I'm looking for a great